All right, I just sent a poll. When you are done, go ahead with your drawings. Go ahead and click yes. All right. It looks like most of you are done, so what we're going to do is I'm going to go through and turn on your cameras so you can go ahead and share what you have. Robert, it doesn't look like your camera is connected, so if you could go down to the bottom left-hand corner, there's a little wrench. Go ahead and click on that for me. Choose whatever web camera you have connected to your computer so that you can share your picture with us as well. That would be great. And if you don't have a web camera, then if you could email me your pictures at the end of class, that would be good. So, Sarah, you are first. In order to share your web cameras, what you guys need to do is underneath the black screen where my video normally comes through, you should see a start button. Go ahead and click the start button for me. And then that should turn your camera on. So Sarah, whenever you're ready to click that start button, go right ahead. Perfect. Hello? Just go ahead and show us your picture, and then make sure you click the talk button so you can talk.
Okay. I do the bacillus and the coccus and the spirillum, and I do some that are in clusters and some that are in pairs and some that are single. Yeah.
Very good. Fantastic. Thank you very much. All right, Gabrielle, you are next. You should see a start button underneath your black video screen now. Go ahead and click start. And then your video should come through. There you are. I'm going to go ahead and click talk. I don't think you clicked talk on your side. Make sure you go ahead and click talk.
I do the coccus and the bacillus and the spirillum, but I didn't label them yet, so I still need to label it.
All right, fantastic. Thank you very much. Alicia, you are next. Go ahead and click the start button underneath your black screen. All righty, make sure you hit the talk button.
Okay. All right. I drew coccus, bacillus, and spirillum. See like that. My coccus, and I labeled one with streptococcus and diplococci and monococci. And then there's my bacillus, and then there's my streptobacilli, my diplobacilli, and there's just my bacilli, just one. And then I did not put any labels on the spirillum because they are only seen in one, So they're just by themselves swaying in spirally ones.
Awesome. Thank you very much, Alicia. And Anthony, if you could go ahead and click on yours underneath the black screen, there should be a start button. Thank you guys all for being such willing participants in class. Remember, part of your grade is participation. So, all right, there you are, Anthony. Go ahead and make sure you click the talk button.
Here's my drawings of the coccus, bacillus, and Sperlium. And I have the three different variants on top.
Very good. Thank you very much, Anthony. And again, thank you guys all for being active participants. Now, the next lab we are going to do, lab number 12, we are going to look at bacteria at different places, either in our house, in our yard, or on your body. Before we do that, I want to share a little video with you guys about extreme bacteria. Now, extreme bacteria are called extremophiles. There are bacteria that can exist in all sorts of environments, from the deep ocean to very acidic, to very basic, to very cold, to very hot environments. This video takes a look at bacteria in each of those different conditions. It's going to give you a good idea, or hopefully some good ideas, of where you can try and collect bacteria for the lab we are going to do. Now, obviously, no one is going to the deep ocean to collect bacteria. However, it may give you some ideas as to places in your house that you may not think of right away. So this video is actually in Jigsaw, so I'm going to give you guys the directions to get there. It's about 4 minutes long. It's going to take you to another website. When you're on the website, you are going to watch the video. When you are done with the video, you are going to come back to class. I am just going to put a poll up on the screen that says click yes when you are back. When you are back, click yes, and then we will go ahead and talk about the video. So on your whiteboard screen right now, make that the largest screen on your desktop for me. There are some icons. The last icon on that purple toolbar says Applications. Click on Applications, which is on the purple toolbar above your whiteboard. You now should see a word that says Extremophiles. Do not click anything. Okay, click yes or no. Do you see the word Extremophiles on your screen? Please do not click to start it yet. Okay, you are looking on your whiteboard. On the whiteboard, you see a purple toolbar. At the very top, you should see icons across the top. The very last icon says Extremophiles. I'm sorry, the very last icon says Applications. If you click on that with your mouse, it should open up and you should see Name and Extremophiles. I just resent the poll. I'm waiting for one answer. Okay, perfect. Everyone can see it. Go ahead and click Extremophiles, and I will see you at the end. Okay, we're waiting for one more minute, so I'm going to give him another minute, and then we will go ahead and talk about the video. Awesome. Well, we are all back. Fantastic. Now to get back to your regular whiteboard, you are going to click on the very first icon that says whiteboard. I'm going to unlock your whiteboards, and I want you to write just a few comments. Actually, we'll talk about it. We won't write about it. What did you guys think about the video? Now, obviously, you're not going to go collect bacteria from anywhere in outer space or from the polar ice caps. But it may give you an idea of where you could collect bacteria around your house that maybe you didn't first think of. Everywhere around your house has bacteria in it, and we are going to come up, hopefully, with some good bacterial samples just from your house. But what did you think about his pictures that showed the spirillum-typed shaped object and the bacillus-type shaped object that he took out of the meteoroid or an asteroid? Anyone have any thoughts on that? Alicia, go ahead.
I thought it was really cool to see how alike but yet different some bacteria were. Like, you could see the similarities, but in a lot of ways they were just so different in how they came from space, and that, you know, these might actually be the fossilized remains of other bacteria out there, and that we may actually have extraterrestrial life, just, you know, not green aliens with big black eyes.
Good job. And it definitely is a thought. I mean, he did say it was a hypothesis. There's nothing proven in what he is doing. He is an astrobiologist. During this class, I'm going to try to expose you guys to some different types of biologists. You know, not the person that you when you think, oh, he's a biologist, he's out there studying the deer population. You know, I don't know how many of you ever thought before that there were actually astrobiologists that studied potential bacteria from space. It's a different type of science that's not what we would consider the normal science. Anyone else have any thoughts about the video? All right, well, let's go ahead and get started on lab number 12 then. Lab number 12 is on page 55 of your lab book. Now, I'm hoping that you guys went ahead and read this beforehand. If you did, fantastic. If you didn't, you were supposed to. So you're going to have to skim along. So if you look at the very top, it says, note, the initial setup for this lab is short. The lab will take many days of observations to obtain the results. What you are going to do today is set the lab up. What you are going to do for the rest of the week is observe what's happening. Bacteria can sometimes be very helpful. Bacteria can sometimes be very harmful. What we are going to try to do is grow bacteria in Petri dishes. So in your lab kit, you should have a Petri dish or a culture dish. Go ahead and take your dish out. Now, before we go ahead and get started, is there anyone who would like to volunteer to turn their cameras on and let us watch you set the lab up? I'm warning you, when we do dissections, that's going to be a requirement. At different points, we're going to turn your cameras on. So does anyone want to go ahead and volunteer today? All right, thank you, Alicia. I'm going to turn Alicia's camera on. And if you could just try to angle the camera so we can see what you're doing, or if once you do a step, you pick your sample up, that would be fantastic. Alrighty, so we can see you. So we are going to start on page 56, where it says, note, please keep the top and bottom of each culture dish firmly closed until instructed to open them. This will keep them sterile and not introduce any bacteria until the correct time. You should have 2 Petri dishes in your lab kit. We are going to start by turning both culture dishes upside down. So the larger, kind of the lid, is going to be face down. Using a Sharpie pen or another type of marker, divide each dish into 4 parts as shown in figure 1. Mark one dish as dish 1 and one dish as dish 2. So go ahead and complete step 1. Okay, you can use a Sharpie, you can use a marker, something just so you can see the letters C, B, T, and M, the division points, and labels dish 1 or dish 2. Step 2. Mark each section as instructed. Mark one section with a C to represent the control. This section will be left untouched throughout the experiment. B, to use for bacteria collection before you brush your teeth in the morning. T, to be used after you brush your teeth with toothpaste. And M, to be used after you take the additional step of using mouthwash. Now, if you do not have mouthwash or do not use mouthwash, what you are going to do is after you brush your teeth with the toothpaste, you are going to rinse your mouth out a few times with water. So if you have mouthwash, fantastic. If not, you are just going to rinse your mouth a few times with water. Step number 3. Bacteria are living organisms and so need certain nutrients to grow. To make the nutrient medium for the bacterial growth, you will need to do the following. We need to boil at least a cup of water. Okay, you can either boil it or you can microwave water till it gets really hot. It is definitely best if you are boiled, or if not you, if the water is boiled. You then are going to do steps B and C. You will need the following supplies: your gelatin, your bouillon cube, a small bowl for mixing, and 2 measuring cups. You are going to put everything together, steps 1 through 6, and then we are going to put this mixture into the refrigerator. When you put the mixture, once it's in the dishes, in the refrigerator, you are going to put the lids facing up. So what I would like you all to do right now is go ahead and complete step 3 of this lab. Okay, and we'll watch Alicia as she does part of this. And I know you guys are going to have to go boil water and stuff, so that is perfectly fine. Hey, if you have any questions, you certainly can ask them. I am here. But if not, just go ahead and click yes when you are done with step 3. You guys are just stopping at 3. You're going to do step 3, A, B, C, and then you are going to stop. So when you get to the end of page 57, you are stopping. If you're having a hard time bouillon cube to dissolve, as long as it partially dissolves, that is fine. The bouillon cube is really just for nutrients, and your bacteria aren't going to need a ton of nutrients, so you should be all set. Okay, if you're looking at Alicia's screen, she's doing a great job. She has her medium in the bowl. She's now pouring it into her Petri dishes. So you're a fantastic job there. Everything is now made. Once you have the beef bouillon and the gelatin and you have it all mixed together, put it in the bowls and then cover them up. Your goal is to keep them covered as long as you can. So you want to put the covers right on. Perfect. Fantastic. And then just leave them. Okay, you don't want to take those covers off again. Now what you are going to do is you are going to take them and you are going to put them in your refrigerator. This will solidify them. When you put them in the refrigerator, make sure you tell everyone else in your family that do not touch them. They really are only going to take about an hour to get hard, so they do not need to be in there forever. Once they are hard, you can go ahead and take them out and put them on a counter. Do not put them in direct sunlight, but you can put them in your bedroom, on a counter, or you can leave them in the refrigerator until tomorrow morning. It is completely up to you. The only job of the refrigerator is to solidify the gelatin mixture. So it looks like you are all complete. Alicia, again, thank you so much for letting us watch you complete the lab. I'm going to turn your camera off and mine back on. Now what you are going to do is either put them in the refrigerator now or wait until class is over. And we are going to do the rest of the lab tomorrow morning. The best time to do this is first thing in the morning because there is lots of bacteria in your mouth when you first wake up. Before you eat anything, before you brush your teeth, nothing. In the morning, you are going to do step 4 all the way down to step 11. Now, it asks you to do this on 2 Petri dishes, to do the same swabs with your cotton swab. I would like you guys to do something a little bit different. I would like you to do 1 Petri dish exactly how they are describing it here. Do your dish 1. I would like you to do dish 2, testing 4 other things around your house. So, for example, one when I was teaching in a school that we always used to do was our computer mouse. Just take a cotton swab, dampen it with a little bit of water, and touch the area you would like to test. So let's say I'm going to do my computer mouse. I'm going to swab my computer mouse. Then I am going to take and I'm going to make a smear in my dish 2 in one of the letters. So let's see, I make it in the C. Then on day 1, I'm just going to write computer mouse. And let's say my B section is going to be the telephone, my cell phone. I'm going to write it there, make the swab. You can do 4 things around your house. If you want to do something outside, that's fine. Say you want to test the type of bacteria that is on the car handle. You can go ahead and do that as well. Same thing, you're going to wet the Q-tip, touch whatever you would like to test, and then touch the Q-tip to your Petri dish. Then every day for the next 8 days, or every day starting tomorrow, and for 8 days, you are going to observe the bacteria growth in your Petri dish. If you can see colonies, like we talked about today, the cocci, the bacillus, the spirillum, you are going to write bacilli bacteria. If you can't, but you can see an orange color, you are going to write that. Ideally, your Petri dish in some sections is going to have what looks like little circular or rod-shaped bacteria. Now, that is ideal. It is not going to happen for everybody. Once you make all of your samples, you need to find a dark place to keep these samples that is at room temperature. My suggestion is a drawer in your bathroom maybe that doesn't get gone in a lot, or a corner of your closet that does not have a window in it. That way you can observe it, then walk out of the closet, open the drawer, observe it, and close the drawer. You do not want it in direct sunlight, and you do not want it somewhere that is too hot or too cold because the bacteria will not grow well. So homework is to make these observations. You do not need to worry about the questions on page 61 yet because you're not even going to have all of our data by class next Wednesday. So you are not turning any of this into me right now. You are simply taking notes every day in table 1 and 2. Honestly, it should take you about a minute a day. It is not going to be time consuming. Your real homework for this week is to choose a lab in your lab book. It can be any lab that we are not going to complete this year. If you look on the syllabus, it has all of the labs we are doing by number. If you have lost your syllabus, that syllabus is handy dandy on the Moodle website. So you are going to pick one lab, you are going to complete that lab, and you are going to write up a lab report. Any lab that we are not doing in class, you are going to turn the lab report in to me for homework. Does anyone have any questions either about the rest of the lab that you need to do or any questions on your homework? All right, well, fantastic. That is where we are going to stop for the week. You guys did a great job. I'm very impressed with you and your drawings and your work today. If you have any questions during the week, give me a call, send me. Otherwise, I will see you next week. Reminder, if you did not do last week's homework, make sure you get it to me. It is 1 letter grade a day late. Okay? Have a great week, and I will see you all next Wednesday.